The scientific method is the source of all scientific knowledge that we have. And generally, the scientific method is thought to consist of 5 different steps. Now, some people will say 3 steps, some people will say 4 steps, but they're all talking about the same thing. They're just breaking it up in different ways. I, however, will talk about 6 steps to the scientific method. And the 6th step that I'm going to give you is not usually talked about by others, so do pay attention when we get to that. Okay, the first step in the scientific method is observation. All good science starts with careful observation. All right, and the best example I can give is astronomy. Okay, so ancient people watched the sky and they watched it very closely. And they noticed that some objects were visible at some times and not at others. They noticed that different objects moved through the constellations, right? And so they eventually were called the planets. So careful observation of the sky gave birth not to just astronomy, but also to a lot of physics. I'm going to go through all these steps again with an example, and I'm going to give you a more down-to-earth example. All right, so after careful observation of natural phenomena, right? So that we're talking generally about science, so we're studying natural phenomena, be it the movement of heavenly bodies through the sky or the growth of plants in your own backyard. So based on those observations, generally you're going to pose a question. Why does the sun rise every morning? Why does the moon rise sometimes? And of course, those are ancient questions and they have long since been answered. Along with that question, so when you pose a question, for example, why are my plants growing so poorly while my neighbor's plants grow so well? That's a good question. So it's tempting to think that you're going to take that question and do an experiment to answer that question. But you're not going to do that way. At that point, you're going to pose a question and then you're going to do background research. Okay, so the saying we have in biology is that an hour in the library can save you days in the lab. All right, before you rush off and do an experiment, you're going to ask your neighbors, what's your secret to getting such good plants? You're going to look in on the internet and ask, how come my plants aren't growing? You're going to do some background reading. All right. So after you've posed a question and you've done some background research, then you're going to propose a hypothesis. Okay. All right. So a hypothesis is a statement. First of all, it's not a question. So it distinguishes itself from the previous step in that you're making a statement rather than asking a question. And that statement, of course, is a potential answer to that question. Or an educated guess. And it's often, not always, but often in the form of an if-then statement. And you're going to see the advantage of an if-then statement in a couple of minutes. And then at that point, after you've proposed a hypothesis, of course, the most important thing about a hypothesis is that it's testable. That is, you can do in an experiment to address your hypothesis. If it's not addressable by experimentation, then it's not a hypothesis. All right, so and at that point, you're going to go ahead and test the hypothesis, and that comes in the form of an experiment. And this is the most difficult step. Okay, I'm going to go through these with an example. And it's going to be a down-to-earth example that we can really do an experiment on, but you'll see that you can quickly get into a situation where the experiments become too difficult to really do. And then the experiment, of course, leads to some data, and you're going to use that data, and you're going to, and then you're going to draw a conclusion. All right, and the conclusion is going to be that either the data support the hypothesis or the data disprove the hypothesis. All right, the data do not prove the hypothesis. They can only disprove it. They can support the hypothesis, but not disprove it. Of course, the experiment can be inconclusive as well. And that, of course, plagues us often in the real world. And once you've made that, once you've come to a conclusion, you're going to ask, again, that conclusion concerns your hypothesis. So at this point, you might go back to the hypothesis and pose a new hypothesis if your hypothesis was proven wrong. On the other hand, if your hypothesis was supported, you might try to formulate a more refined hypothesis and then do more experimentation. And then the sixth step is publication. All right, science is not done in a vacuum. Whatever we get, we tend to tell other scientists about it so that they can build on our work and we can build on their work. All right, so let's go through this with a real world example. All right, so let's start with observation. And I'll use the observation that I just said. My tomato plants are not growing as well as my neighbors. All right, or maybe I have tomato plants planted in different places, but they're growing better in some places than in others. All right, so at that point, I make that observation. I'm going to ask a question. Why are my tomato plants not growing? And like I said, at this point, you're going to do some research. You're going to talk to your neighbors. What's your secret to growing good tomato plants? You're going to look on the internet. You're going to do some reading. What's necessary for good tomato plant growth? And if you do that research, you're going to find that there are 4 things that plants need to grow. Of course, they need sunshine. And how much sunshine they need depends on the particular plant that we're talking about. They need water, as animals also need water. They need minerals. Okay, and the minerals are really supplied in the water, right? The minerals come from the ground and they're dissolved in the water. So this could kind of be one. And then, of course, they need carbon dioxide. Now, you generally know if your plants are getting sunshine, right? Water and water is something you can take care of if you can water them on a daily basis. Carbon dioxide is not something we can control, so that's not really something we have to test. It's in the air around us, and it's either there or it's not. It is there, and there's carbon dioxide for all living things. So so you'll probably, if you do it this way, you'll probably quickly focus on the minerals. And you'll also find, as you continue to do research, that those minerals can be provided by fertilizing. All right, and so from there, you're going to formulate a hypothesis. And like I said, a hypothesis can be in the form of an if-then statement, but it doesn't have to be in the form of an if-then statement. But in this case, let's go ahead and do if-then. If I fertilize my plants, then they will grow better. Simple as that. Now, the advantage of this if-then statement, the advantage of this if-then statement is that it suggests an experiment, okay? So the experiment's kind of already decided for you. I mean, it's pretty obvious the experiment to set up here, all right? And that is grow. So you're going to grow 1 or more plants, preferably more, with fertilizer. And then you're going to have 1 or more plants without fertilizer. Okay, so that's a relatively simple experiment, one that we can approach. Of course, experiments can be much more complex and difficult. Imagine a drug trial. With a drug trial, you want to test out a new drug and see if it prevents disease. So in general, in that case, you're going to enroll lots of people, thousands of people, and split them up, give some of them drug and not give some of them drug. And then look at the outcomes. Look at their outcomes in terms of disease. All right, so let's talk a little bit more about this experimental setup. All right, so what we're going to do, okay, so we have 2 plots of land, 1 plot of land over here, 1 plot of land over here, and in each of those plots of land, you're going to put a baby tomato plant. All right, so in this case, it's going to be plus fertilizer. And in this case, it's going to be the standard conditions. That is, no fertilizer, or what you've done before. All right, so this is the experimental group. Now, heads up here, because I'm going to give you a lot of terminology. This is the experimental group. And this is the control group, the standard conditions, the one that's not being perturbed. Okay, and that leads us to the more terminology. So experimental variable. Experimental variable. All right, and in this case, the experimental variable is whether or not you're giving iron. Excuse me, whether or not you're giving fertilizer, with and without fertilizer. All right, so be careful. Experimental variable, experimental group. And then, of course, we have control variables. And this is kind of the crux of the matter. Okay, there can be one or usually many control variables. So a good experiment varies one thing at a time, varies one thing and keeps everything else constant. We want to see the effect of fertilizer. So we're going to vary fertilizer, but we're going to keep everything else the same. All right, and everything else we're going to keep the same are all the things that we talked about before, right? All the various things that affect plant growth. All right, so what we want to do is we want to make sure that both sets of plants have the same amount of sunshine, the same amount of water. Remember, we're starting with baby plants, so we want to make sure that the initial health of those two plants is the same. They're both good, healthy, young plants. And of course, they're the same species, the same plant, bought from the same vendor, as much as similar to one another as possible. And then we have to, look out for herbivores, rabbits and squirrels that might eat your plants. You don't want one set of plants to be subject to herbivory while the other one is not, because that would throw off your experiments. All right, so you can see there's always many control variables. In this situation, it's, again, a very doable experiment. These things are relatively easy to control because if we just put these plants near one another, they can't be too near one another because one's getting fertilizer, the other's not. But if we put them near one another, then they're pretty much guaranteed to get the same amount of sunshine and water. And they're going to be exposed to the same herbivores. And of course, we can look at the plants and make sure that they're similar in health when they start. So this experiment is very doable. But again, these experiments can get very complicated. Think about the example I gave before with the drug trial. If in a drug trial, we want to have the people who get the drug versus the people who don't get the drug, or people who get the placebo. We want those people to be as similar as possible. And that's very difficult because no two people are alike. People are very different from one another. They're different in terms of age, their gender, their difference in terms of their health history, different in terms of their genetics. So there's really no way to control for experiments that we do with humans. So we kind of get around that by using lots and lots of people. All right, so drug trials are usually done on thousands, if not tens of thousands of people. And then once a person enrolls in a drug trial, they are randomly assigned between the drug group and the placebo group. And that way there's no systematic bias. You're getting all younger, healthier people in the drug group and not in the control group. That would really throw off your results. So you don't want to be any systematic bias like that. So you randomize them and you use lots of people, hoping that the differences between people will even out between the two groups. Okay, so that is our experiment. And then, of course, as we do the experiment, we're going to collect data. Data or the results of the experiment. And of course, the results can take many forms. All right, so data can be qualitative, or it can be quantitative. Quantitative, meaning that it involves numbers. Okay, so qualitative, how does the plant look? What's the color? Is it a nice healthy green color? Those are qualitative measures. Quantitative measures is how tall are they? How much mass have they accumulated? How much fruit have they given rise to? All right, so, and of course, once we get quantitative data, generally we're going to present that quantitative data in the form of tables and graphs. All right, and we're going to learn about those in a future lab. All right, so data can be qualitative, quantitative. And then the other thing we should talk about is dependent versus independent variables. Okay, so the independent variable is the thing that you manipulate. All right, in this case, we're manipulating fertilizer, well, whether or not we have fertilizer. So that's going to be the independent. All right, and then the dependent variable is the one that you measure. All right, and that's going to be these things that we talked about before: the height, the weight of the fruit, and so forth. Okay, so think of it this way: the height of the plant depends on whether or not there's fertilizer. Okay, so that the dependent variable depends on the independent variable. And when you do go to graph this stuff, the independent variable goes on the x-axis because it's independent and doesn't need any support, whereas the dependent variable goes on the y-axis, and that is supported by the x-axis. All right, so now we've set up our experiment, and thankfully it's a relatively simple one. And we've gone ahead and collected data. So let's take a look at a hypothetical outcome from such an experiment. And okay, so here's the hypothetical outcome of our experiments. And remember, this is the one with fertilizer, the one without fertilizer. And from this, we can draw a conclusion. And the conclusion is the data supports our hypothesis. That is, when we add fertilizer, the plants grow bigger and healthier and stronger. All right. So now if it had come out the other way and we didn't see a difference between that, then our hypothesis would be disproved. And at that point, we'd go back to our hypothesis and come up with a new one. Maybe it's not the fertilizer. Maybe it's the water. Maybe it's the sunshine. Maybe there's some other factor that we're not thinking of. All right, so, but even in this case where the data does support our hypothesis, we still can go back and now make a more refined hypothesis. Okay, so the fertilizer helped. Is there a particular component within the fertilizer? Is it the iron? Is it the calcium? Is it the potassium? So either way, you do go back to your hypothesis and either toss out the hypothesis and come up with a new one, or come up with a more refined one. And then, like I said, the last step is to publish our results. Now you can go ahead and do this experiment, and you might have fun doing it and you might learn something, but if you're not publishing it and telling others, then you're not doing science. All right, so the reason that we publish is that so other science can look at it, other scientists can look at it. And a scientist in this case is anyone willing to read your publication. All right, and then those scientists can look at your work and they can criticize it if they don't like the way you did it, you overlooked something, you didn't do a proper experiment. They could praise it, saying, oh yes, you did a very good experiment and your results are very solid. But in either case, it's out there and other scientists can build upon it. All right, so we have a picture that comes from fiction, be it books or movies, but the picture is of a lone scientist working in a lab on their own in order to come up with some miracle cure or some great advance for civilization. That's not the way real world works. In the real world, you have a group of scientists working together and they make some small advance and they publish that small advance. Then another group of scientists working elsewhere sees that work and they build upon it. They make another small advance and then it goes back and forth like that. And over the course of years or decades or even centuries, science advances. And that, of course, is what's given rise to the very scientific and technological world that we have today.